Hello, my name is Harry Forsyth with Analytik Jena. Welcome to our exhibitor tutorial at SLAS 2021. Analytik Jena, the destination for lab automation. By using excellent liquid handling and a variety of automation systems. Explore #HowToFelix and learn how you can automate your workflow. #HowToFelix. Our story continues. Our automation solutions can be adapted to a broad range of applications. Today we have the pleasure to introduce you to two speakers who are at the forefront of their research and will be sharing with you how our CyBio FeliX helps them achieve their goals. First we have Yongbao Wang, Ph.D. from Jewish Health, presenting on high-throughput Covid19 PCR testing with implementation of CyBio VARIO and CyBio FeliX. Secondly, we have Eric Parigoris, a Ph.D. student at the Wallace H. Coulter Department of Biomedical Engineering at Georgia Institute of Technology, under the mentorship of Professor Shuichi Takayama, presenting on cancer cell invasion of geometrically inverted mammary organoids using the CyBio FeliX. Feel free to use the chat feature to ask your questions, and we post answers after the presentation. You can also speak to our specialist anytime during the SLAS show at our booth. Thank you. High-throughput Covid19 PCR testing with implementation of CyBio VARIO and FeliX. Dr. Yongbao Wang has more than 20 years experience in the biotechnology and clinical diagnostics fields, currently serving as the Senior Director in Advanced Diagnostics Labs at National Jewish Health. Dr. Wang oversees the Department of Strategic Innovation, responsible for new clinical assay development and workflow automation. He previously served as Senior Director in Assurix Health, then part of the Neuroscience Division of Myriad Genetics, where he oversaw assay development, platform adaptation, and workflow automation. He was also a Scientific Director of R&D in Anatomic Pathology at Quest Diagnostics, and a Senior Scientist where he developed more than 60 new assays in Quest Diagnostics, including LookAdvantage. He has a broad experience in lab management and new technology translation into clinical labs, with multiple publications and 4 issued patents.
So National Jewish Health is a hospital with more than 120 years of experience in the patient respiratory care. So we have more than 120 plumberists. So we have a clinical research division, academia research, and advanced diagnostic labs. So in advanced diagnostic labs, we routinely run all kinds of lab-developed tests as a reference lab for our hospital service. We also develop new assays for CRO and for biopharmaceutical and biotechnology companies. So we are CAPA and ISO certified. We are a GDPR compliant laboratory. In advanced diagnostic laboratories, we have complementary immunology, microbiology, and microbacteria. That's our TB lab. Also, we have a small PK lab, do pharmacokinetics, and so also we built our virology lab last year as COVID-19 testing. Strategic innovation is our development division. So we develop new assays for our reference lab. Also, we develop new tests for biotechnology companies and biopharmaceutical companies. So you can see here our expertise everywhere, from immunoassay to flow cytometry to the cellular function assay, sequencing molecular assay, NGS, also the HPLC and TB, NTM, and drug testing. Today, I'm going to talk about our journey on COVID-19 PCR-based testing in 2020. So we validated our CDC COVID-19 testing back in March. That's very early, so in the middle of March. So that's a 4-well singleplex, later on reduced to 3-well real-time PCR assay targeting 302 COVID-19 specific targets, has on-ASP as internal control. Then we validated another assay in April that is one-well multiplex real-time PCR assay has a 3 SARS-CoV-2 target on protein and S protein, 4 color, 4 dyes. So we also developed and validated our third COVID-19 assay that is based on the reverse transcription PCR and MERD12 technology in late May. We submitted for FDA EUV approval and get approved in October. So it's a one-well reaction. We put 5 SARS-CoV-2 targets there, plus 1 internal control and 1 external control. So immediately, just as other hospital lab, we are facing another challenge that is scale up. Before COVID-19, we are just like a hospital lab. We do a reference testing and we do CRO testing, but we are not that really high throughput lab. So the challenge we are facing is, okay, how we can run samples from every day 100 to 200 to the 1,000, 2,000, even 3,000, 4,000. So that's a real challenge we are facing. So luckily, very soon we set up our, the high throughput workflow with Cybervalue and FeliX as a core components for this whole workflow. So based on 384 format. So currently we build our workflow. We can test 6,000 samples, even more every day for the COVID-19 PCR assay. So this is really something we haven't done before in this hospital and in this lab. So we are so proud of it. So let me first briefly introduce our third assay that is based on the MERD12 technology. So also our automation is really focused on this assay. That's because of multiple reasons I will talk about later on. So this is a comprehensive PCR-based COVID-19 test. It's suitable for self-collection as well. The FDA require to do self-collection, you need internal control like on ASP. One-well reaction, 5 SARS-CoV-2 targets, 1 internal control, 1 external control. So the chemistry is based on the reverse transcription PCR. We are detecting a virus RNA. Then detection, we use the MERD12 mass spectrum. So small reaction volume, 5 microliter or 10 microliter PCR. It didn't require multiple steps. So PCR after that is sharp clean up and target specific single prime extension. The total take about 3.5 hours. Detection is a mass MERD12. So that is to detect the extension product by MERD12. So there's multiple reasons why we chose this platform. First, we avoid the reagent supply bottleneck. During COVID-19 area, so we experience the every supply shortage from the 96 vial plate, 380 vial plate to the reagents. So secondly, we have much lower cost because without labeling problem and time. So this assay has a higher assay tolerance because it's not based on real-time PCR. It has a higher assay sensitivity. So this assay can equally detect the SARS-CoV-2 variants, non-SARS-variant COVID, such as B117, 501V2, and cluster 5. So also this assay is expandable. So we are adding more targets can differentiate this variant into the assay. We built our high throughput COVID-19 PCR testing workflow. CyBio, VARIO, and FeliX are one part of really core elements for this automation workflow. From this slide, you can see from RNA extraction, we use FeliX 96 1250. Then we RNA loading from 96 to 384, we use 96 250. So then after PCR, we use Cybervalue 384 to add a sharper mask mix to PCR plate. And later on, we add the extension mask mix to the sharper product plate. So this 3 instrument is really a core our workflow. First, let me start with RNA extraction. FeliX 96 1 to 50, so plus inu prep viral DNA RNA kit is our one RNA extraction platform, because we have multiple platforms in-house. So this instrument is very flexible. So one thing I want to particularly point out is this platform has a high recovery of viral RNA. That's kind of surprising for us, actually. So we got the highest LOD from this platform. So after RNA are extracted, so then we use FeliX 96 to 150 to do a RNA loading. So that is from 496 vial plate load RNA to 1384 PCR plate. So the loading volume is 5 to 10 microliter, and this is really easy to script, fast, just take a few minutes, accurate and very consistent without any contamination. After PCR, then we use the Cybervalue 384 to do a post-PCR processing. So Cybervalue is really a central part of our workflow. So when finished the reverse transcription PCR, we add 2 microliter of SAP to do cleanup. Use the Cybervalue, then move back to thermocycler to do an incubation. Then after that, add 2 microliter of extension mask mix to the product. So you can see here, Cybervalue is a central part of this 384 workflow. One push button operation, you can finish from transfer to the mixing. Then move back to thermocycler. 1 hour later, we'll be ready for the detection. From our experience, Cybervalue 384 is the best choice on handling a small volume of the liquid, so especially on 384 platform. So it has a high accuracy and precision, and it has a switchable head, is multiple purpose. You can switch out anytime. Transfer 384 vial PCR product in minutes. So also automatic pipette. You know how hard it is to mix this small volume in these small vials. So specifically, I want to talk about why we chose CyBio FeliX. So this is a very small footprint liquid handler, about 1/3 of other liquid handlers. So get a dual deck design is very unique. I never seen something like this before. Lightweight, you can easily move in and easy programming, plug and play if you use the Analytik Jena kit. High accuracy of the transfer is eco-friendly, you use less tips. So also we are considering post-Covid19 area, so we can use this liquid handler to do other assays. So here we are using this platform to do RNA extraction and the RNA loading. Thanks for attending this webinar. I'm glad to take any questions.
Cancer cell invasion of geometrically inverted mammary organoids with the CyBio FeliX. Eric Parigoris, Ph.D. students in the Wallace H. Coulter Department of Biomedical Engineering at Georgia Institute of Technology under the mentorship of Professor Shuichi Takayama. Eric received his BS in Mechanical Engineering from Carnegie Mellon University in 2016. Upon the completion of his undergraduate degree, he studied at ETH Zurich on a Whitaker International Fellowship. Eric's research interests include organoids, mechanobiology, and organ-on-a-chip systems.
Hello everyone, my name is Eric Parigoris and I'm a PhD student in the Biomedical Engineering Department at Georgia Institute of Technology in Shu Takayama's lab. And the title of my talk today is Cancer Cell Invasion of Geometrically Inverted Mammary Organoids with the CyBio FeliX. So I'm sure many of you are familiar with them, but I wanted to give a brief overview of organoids, which are essentially 3D balls of cells that recapitulate the native architecture found in many organoids, ranging from the breast to the kidney to the lung to the intestine, brain, among others. And because of their potential, they were actually voted Method of the Year in 2017 for their opening up new possibilities for answering a range of biological questions. So there's a wide range of applications ranging from disease modeling to drug discovery to gene editing to precision and personalized medicine. And in our lab at Georgia Tech, we're particularly interested in disease modeling. And some of the challenges with organoids that we aim to tackle are making uniform, reproducible organoids in a high throughput manner. Another historically difficult challenge is accessing the apical or the luminal side. So you have this enclosed space and it could be very difficult to access. So this is one of the engineering challenges we're really interested in. While there are many different approaches and methods to grow organoids, one that has been very successful in our lab is called the hanging drop technique. So hanging drop culture essentially uses surface tension to hold a drop of media on the bottom of a plate that we have custom made in our lab. So they have a through hole and the drop hangs at the bottom. And you can see in panel B, we just put food coloring in to help visualize them better. And we don't use every well, we use every other well. So essentially this is 192 wells of a 384 well plate to avoid the droplets merging together. Because they're hanging under the well, you can imagine it's very sensitive to any slight bumps, jerking motions, or disruptions. So how are the organoids or sometimes spheroids forming? So essentially you pipette the media with a cell suspension in the through hole, and the cells go to the bottom, and over time they can aggregate into an organotypic structure, and they just simply sit at the bottom of this droplet. As you can imagine, these cells are very the droplets are sensitive, and there's 192 in a plate. So this is where the CyBio FeliX comes in, which I'll talk about in detail in a little bit, but we use this to maintain the media plates over long periods, we maintain the plates over long periods of time. And another challenge of these plates is that because the droplets are just sitting at the bottom, evaporation can be an issue. So we place distilled water on the bottom, and we fill gauze pad on the side troughs and also place distilled water on that to prevent evaporation. But on the other side, because it's open to the surrounding air, there's very good air exchange and gas exchange in these plates. So what does it actually look like? So over here you can see a picture of the drops just hanging, and this is taken from the underside of the plate. And you can see in each of them there's a little white ball, and that's actually the organoid sitting right there. And the droplets are typically 25 microliters. And you can imagine placing the pipette and positioning it can be very, very difficult. If you push it in and it gets wedged, you can pick up the whole plate with the pipette, and you can ruin your plate. Or if you bump it, or by accidentally pick up too much, you could aspirate your organoid. So there are a lot of considerations for successfully handling and maintaining these plates and the organoids in the plates. So as I mentioned, they're very delicate and sensitive to physical perturbations. Because the droplet is just sitting there, if you take up all 25 microliters, you'll simply aspirate the entire organoid. So we do 1/3 media changes. So we remove about 8 to 9 microliters at a time and replace them, and we consider that one media change. Because we only can do 1/3 at a time, and we have to do multiple consecutive ones, and because there's so many wells, it's very, very time consuming. And one of the biggest limitations is if we introduce a drug and we want to completely remove it or have about a 100% media replenishment, we have to do 7 to 8 consecutive media changes, which takes hours on end, and it's not really realistic. It takes half of a day to complete that. So this is where the CyBio FeliX comes in, and it's been very, very helpful and allows us to maintain, as a lab, maybe 10 to 15 plates among multiple lab members. So one of the advantages is there's minimal plate handling and no manual pipetting. So instead of using a repeater or multi-channel pipette, which could be very difficult to align in multiple holes at once, or just be very tedious and time consuming, we just put the plate down, we press go, and then after a few minutes, the media change is done. Another really important consideration is there are no sudden movements. The FeliX is very, very smooth, and we can control all of the deck speeds very precisely and slowly. So if the whole deck were to jerk, all the droplets could fall down. Or if the pipette speed of aspiration was too quick, it could suck up the organoids. So we can control very slow pipetting speeds as well. Another highlight of this machine is it has very precise tip positioning. So keep in mind that the pipette tips have to align with 96 wells at once. And so if it goes too far down, it can aspirate the organoid. And if it doesn't go far enough into the hole, it won't aspirate media. And so you can control the X, Y, and Z position very, very precisely. And as a result of this precision, there's rarely a loss of a droplet and rarely organoid aspiration, meaning it aspirates the organoid and we lose it. We can have very quick media changes, so it takes about 2 minutes to perform a media change. Keep in mind it's only 96 wells, so it has to go in 2 positions to get all 192 organoids, and then we repeat this cycle 2 times, and we consider that a routine media change. And now we are able to do the 7 to 8 media changes for a full replenishment in about 15 minutes. And that's not 15 minutes of active work. We can press go do another task, and come back in 15 minutes, and it's completed. So here's a quick little video showing a media change. So first, what's going to happen is we're going to remove 8 to 9 microliters in 96 of the wells. And you could see it moves very, very slowly. So if something is caught, it doesn't lift the whole plate. And then it dispenses this into our waste container, and it performs a blowout there, because if we were to blow out in our droplets, it could push them through. Now there's a feeder plate, and it's going to take up maybe 9 to 10 microliters. Remember how I said there was a little bit of evaporation? Adding a little bit extra each time mitigates this evaporation. So then it will go do the same thing for the other 96 wells, and then it will repeat that whole process again, and that is a whole routine media change. So because of this technology, we're able to maintain, like I said, many, many plates at once. And so what are some of the interesting questions that we can ask? So our lab is very interested in mammary organoids. And you can see in the top panel over here, we've developed an organotypic system, which is actually made from a cell line. We use immortalized MCF10A cell lines. While organoids are historically generated from stem cells, we call these organotypic structures because we have expansion over a 16-day period that leads to very large, about 1 millimeter in diameter, hollow structures. And importantly, they resemble the native architecture found in the breast. As you can see, it mimics an H&E picture over here. Another reason we find this platform very powerful is because there are multiple lineage markers that can be seen in the bottom images over here, and also multiple stem phenotypes found in these organoids. And finally, this platform is high throughput and also reproducible, and creates uniform organoids from well to well, which are some of the challenges that many organoid models face. More recently, we looked into a little bit about the morphology and formation of these organoids, and we found that over time, we seed these cells in the hanging drop. Over time, the cells form around a matrigel, and we use a dilute amount of matrigel that pre-gels, and the cells form around it. And as a result, you can see from an H&E picture, there's pink hues in the middle, meaning matrigel gets trapped inside. With a lot of other organoid cultures, it's placed in a matrigel dome with the matrigel on the outside. But in this model, the small amount of matrigel we use is partially gels and gets entrapped in the core. And as a result, the cells sense this basement membrane signal and orient their their polarization in the opposite manner. So typically, the apical side faces the lumen and the basolateral side faces the outside surrounding media. But because of this matrigel entrapment, we can see the basolateral side faces the basement membrane, and the apical pole actually faces the surrounding media. So we are calling these geometrically inverted or basal in, apical out organoids. And to further confirm this morphology, we performed immunostaining for laminin 5, which notably is not just in the matrigel. This is cell-produced, and we can see it's localized to the inside of the organoid. So this is, overall, the cells are sensing this basement membrane cue inside, and then they deposit their own basement membrane. And we further show that integrins are localized on the inside, further confirming this inside-out morphology. Now you might ask, why is this important? Why is this exciting? And as I mentioned before, our lab is really interested in vitro disease modeling, and one disease of particular interest is ductal carcinoma in situ, or DCIS. So looking at the anatomy of the breast, there are two main components: the lobe, and the lobe produces the milk, and the duct. The milk travels through the duct to exit through the nipple. And over here, we see an image of a normal duct, which is hollow, whereas in DCIS, we have abnormal growth inside the duct. And this is a gradual process. You have a little bit of hyperplasia that becomes atypical, and then it transitions to DCIS. And then sometimes, in some of the cases, it transitions to invasive ductal carcinoma. We don't have any good metric of knowing when cases progress to invasive carcinoma or when they remain indolent and remain confined in the basement membrane. So it's very interesting to study this disease, and there's still a lot to be learned about it. Now let's pretend that this duct is an organoid. You can see how there are cancer cells inside. This is very difficult to experimentally achieve. It would take micro-injection, so taking a needle with the cancer cells, poking the organoid and injecting the cancer cells, and then waiting over time if they will invade through the basement membrane and expand outwards. But this is a very, very technically challenging and low throughput method. So this is where our organoid model comes into place. Because our organoids are geometrically inverted, the ductal space or the luminal space is now represented by the outside media. So remember, these organoids are sitting in a hanging drop. The outside space represents this duct region. So now we can simply just pipette cancer cells into the surrounding media, and we can assess invasion inwards over time. And the downstream analysis becomes very easy because we could just perform histology or confocal imaging to assess cancer invasion inside instead of, the challenging attempt of injecting cancer cells inside and assessing invasion outwards. So what does this look like? In panel B, we can see over a period of a few days of invasion, this is a histological section showing that the cancer cells are able to invade inside. And panel C is actually we performed clearing on the organoids and then confocal microscopy, just showing another example of imaging modalities to confirm that the cancer cells can invade inside the organoids. And we had a very interesting observation. So panel D is an early stage organoid, maybe at day 7 or day 8, and panel E is a more mature organoid at day 16. And you can see a very big difference between the laminin 5 or the cell-deposited basement membrane, where it's kind of disorganized at an early state, and at a later state, it has a nice, very clear and distinct formation that's almost continuous throughout the whole structure. So how does a different basement membrane formation affect invasion? So in panel F, you can see that when you add cancer cells, when there's a not fully formed basement membrane, you can see in the gray there's a very, very high percentage of invaded cells. Whereas when the cancer cells are added at a later stage, when the basement membrane is more mature, you could see a much lesser extent of cancer cell invasion. This shows that, basement membrane maturity is very important for assessing whether or not the cancer cells can invade. And keep in mind, this is just a model, but we find it very powerful to to better understand DCIS and using it to assess conditions that may or may not allow cancer cell invasion. And with that, I thank you for your attention. Here's my contact information, and I'm always happy to answer any questions.
Thank you very much for joining us. Visit our booth to speak to our specialist directly. We will be posting answers to your questions shortly.